Testing Mas Herman, apakah suara saya masuk? Testing, apakah suara saya masuk?
Masuk.
Oke. Selamat datang Dr. Asep.
Yes, I hear you. Wait a minute. I will setting. Itu belum bisa mas. Untuk pakai sini saya. Mungkin. Ya, ini ada room gitu. Di sebelah sini. Di partisi. Ada kemal ya? Ada kemal juga. Ini. Oh ini. Iya. Iya. Apaan? 25 apa?
Tes.
Tes 1, 2, 3, 1, 3. Are you ready? Selfie.
Yes, Dr. Asep. Wait a
I stop.
setting for the background. Okay. Please kindly wait a minute, Dr. Asep. We'll soon starting the event this conference. Ya, sudah kita mulai, Dok?
Assalamualaikum warahmatullahi wabarakatuh. May peace be upon you.
Good morning, ladies and gentlemen.
Tabipun. The Honorable, all dear guests and all the participants.
Let us begin by giving you a warm welcome.
Today, we will continue our special event,
International Conference on Agromedicine and Medical Science 2020, Day 2. Let us introduce ourselves. My name is Taufik Hidayat.
And my name is Fasya Azara.
And we will be your master of ceremony today. It's such an honor for us to welcome you here.
On this special event, we have several agendas as follows. First, seminar by Dr. Asep Sukohar, SKET MKes, as keynote speaker.
Followed by seminar by our invited speaker, Professor Dr. Zaili Nurrahman.
Dr. Taweporn Gedaram as second invited speaker.
And our third invited speaker, Dr. Miroslava Rizova.
And the last is closing by Master of Ceremony. Moving right along to our main event, seminar by our special guest, by Dr. Asep Sukohar, SKET MKes.
And it will be moderated by a lecturer from Faculty of Medicine, Lampung University, Ms. Selvi Rahmawati, SSE MSc. But before we start the session, I'm gonna read her profile first. Ms. Selvi Rahmawati education background. She was graduated from Science of Faculty of Biology, Universitas Gadjah Mada, and graduated from Master of Science of Basic Medical Science and Biomedics, Universitas Gadjah Mada, Histology and Cell Biology major. And for her achievement, she was Indonesian delegation in summer research program in Tsukuba, Japan in 2016. And also, she is a research assistant on Indonesia Medical Education and Research Institute, Faculty of Medicine, Universitas Indonesia. So for Ms. Selvi Rahmawati, please welcome.
Thank you for the time, Mr. Taufik and Ms. Fasya. Good morning, ladies and gentlemen, to the honorable keynote and invited speakers, distinguished guests, fellow students, and all participants. Welcome again to the second day of the first International Conference on Agromedicine and Medical Sciences, held to celebrate the 18th anniversary of Faculty of Medicine, Universitas Lampung. Today, it's really an honor for me to be your moderator for today's keynote session, and it's really an honor for me to have the Honorable Dr. Asep Sukohar Estet MKes as our keynote speaker from Universitas Lampung. Good morning, Dr. Asep.
Morning.
Thank you, Dr. Asep, for giving us your time today due to your crowded activity. Okay, today Dr. Asep will be sharing with us an expert talk entitled Anti-Proliferative Activity of Jatropon and Curcumin B from Jatro Fakultas on Hepatocellular Carcinoma PLC5 cell lines. Okay, but before we begin, may I introduce our keynote speaker? So, Dr. Asep Sukohar Estet MKes is a head lecturer in the field of pharmacology and also a candidate professor of Faculty of Medicine, Universitas Lampung. He is now also the vice rector of General and Finance, Universitas Lampung, and also the president on Indonesian Doctor Association in Lampung Regional. Dr. Asep is an expert in the field of pharmacology, molecular biology, medical, legal, and medical science. He got his doctoral degree in law appreciation and master's study graduation from Pajajaran University. Okay. Dr. Asep also got additional education in Australian Works Fellowship, School of Medicine, Flinders University in December 2016, and also participated in Sandwich Lake program from Ministry of Higher Education in the field of pharmacology, Florence University, Italy, from September to December 2012. His research now is mainly focusing on the anti-cancer compound from natural resources, especially. Okay, that was really an awesome curriculum vitae and a brief quick introduction of Dr. Asep. Okay, before we begin this session, let me briefly explain the rules of this session. So in this session, we will have in total 40 minutes. And Dr. Asep will give us a talk for about 30 minutes and followed by 10 minutes discussion on PA session. For the participant, if you have question, you can ask by typing the question in the chat room, or later after the presentation, you can click the icon of raising hand, and later I will pick you to give your question to Dr. Asep. And don't forget to mention your name and institution. Okay, so ladies and gentlemen, without further ado, I would like to invite Dr. Asep Sukohar Estet MKes to the keynote session. To the Honorable Dr. Asep, please, the time is yours.
Okay. Thank you, Selvi, for this time, and I will begin for my presentation. Okay, thank you for your time, Selvi. The Honorable our rector and the Honorable for Vice Rector and Dean Faculty of Medicine, and the committee, the moderator, and my colleagues in Medical Faculty, Lampung University. Thank you for your time. This is for maybe more international presentation. This is my title, Anti-Proliferative Activity of Jatropon and Curcumin B from Jatro Fakultas on Hepatocellular Carcinoma via C5 cell lines. This is my research. Our research is began from the 2010 until 2020. And then we still constant on the active conform from plant in our country. and we're looking for the active control for cancer. Uh, this is the introduction. Cancer has become a serious health problem in the world, continuously increasing prevalence over years, increasing and increasing every year. International Agency for Research on Cancer in the journal in 2018 reported 18.1 million new cancer sufferers, with mortality rate was 9.6 million. Hepatocellular carcinoma are the sixth high prevalence cancer and the fifth of the cancer related to death, with dominantly by the Asian race. WHO was estimated more than 1 million patients will die caused by the liver cancer in 2030. The prevalence of cancer in Indonesia is also reported increase from 1.4 per 1,000 population to 1.8 per 1,000 population in 5 years. Hepatocellular carcinoma is the second high prevalence cancer by 12.4 per 100 million population and mortality rate 7.6 per 100,000 population. This is the introduction about the cancer. The big problem in our cancer, that is the treatment. We are developed in treatment with the activity compound from the plant. However, there are no clinical satisfactory therapy result with the chemistry or the activity compound from the plant. There are current regimens still have several issues about the side effect and high cost of the therapy modalities. We know that the price of the drugs for cancer is very expensive, and BPJS Kesehatan in Indonesia is not covered about it. This is the problem. For many years, traditional medicine have been used as a medical treatment, especially in developing countries like in Indonesia. There are about 40,000 plants in the world, and three third reported founded in Indonesia, with estimate about 950 plants have utilized as a traditional medicine. We call in Indonesia that is a herbal medicine. We call jamu and the other. Herbal medicine are being considered as one of possibility of strategy against hepatocellular carcinoma. Jatropha is a multiple purpose plant with some potential medical uses. Number of the study conducted to analyze medical properties compound, phytochemistry, and its pharmacology as the anti-cancer. Jatropha was known a rich terpenoid as its active compound, that is one of the active compound from the Jatropha curcas. Some of terpenoid compound from Jatropha such as jatropone, sprucenol, and jatropion have exhibiting effect as anti-tumor against P338 lymphocytic leukemia. And then Kurkosen B exhibiting effect as anti-invasive agent called angiocarcinoma cell. Some study conducted to analyze Kurkosen B as anti-cancer is like cholangiocarcinoma cell line, chronic myelogenous leukemia cell line, human non-small cell lung carcinoma cell line, human epidermal carcinoma cell line, human hepatocellular carcinoma cell line, or HEP3B, we call it, and human breast cancer adenocarcinoma cell line. We have analyzed Jatropon in other study. Again, hepatocellular carcinoma HG21886 cell line. This is our concern. Another study to access as it cytotoxic effect. It's like in lymphocytic leukemia or B388 and KB cell line and fibroblast cell line and the other cell line leukemia. The method of this study, there are we culture of PLC5 cell lines. That I tell you about this cell line I get from the Tohoku University in 2012. And I cultured it until now. We put on the Dharmesh Hospital until now. That if you interact with this cell line, I suggest you to contact me and I will suggest you to conduct it in Dharmesh Hospital in Jakarta. Especially for this study, the study was conducted in March 2016 until May 2017 as a research, and we done it in the Dharmesh Cancer Hospital in Jakarta. The PLC5 cell lines, that is culture, uses DMEM from Gibco. And then we harvest until 80% and make sure about the confluence. Because if not reach until 80%, that is maybe your research is not perfect. And after 80%, we treated it with the Kurkonsumbi and Jatropon. Okay, we continue. And then we calculate about the IC50 by the Finney or wheel or Litchfield wheel cushion, and this is the method we use for it. And then we calculate about, or we done with the MTT-SA, and then we calculate about the cell death and the fiber cell. And then we make the table about the fiber cell compared with the dead cell. This is the table 1. We show you about the 5 concentration. The 5 concentration with the Kurkosen B. And the table 2, we show you about the table about the Jatropon. With the 5 concentration, we show you about this graphic. And then if you concern with these lines, that the concentration about the Kurkosen B is like this, and not linearly with the line, this line. This is the value of R2 that is 0.9306. In low doses IC50. If we compare with this table, in table 2, is similar with 5 doses. And we calculate, and I show you about this graphic. If you concern with this graphic, this is the line, and this is the IC50 or concentration of jatropon. Jatropon and curcosunbi in this study is more or less similar. Yeah, that is 0.9903, and we compare with the jatropon is similar but different. Yeah. But if you choose with this two active compound, I suggest you use the jatropon in hepatocellular carcinoma PLC5 saline. If we tell you about in the hepatocellular carcinoma with the other type, it's like HG21886. I think this is different too. Okay, we will continue. This is the result I show you in A and B. Jatropon, curcosun B, that is R2, R quadrat, that is 0.9306, and jatropon 0.9404. By using log calculation, it was known that the ICPP of curcusin B and jatropon value that is 0.2826 microgram per milli, and or 9.57 micromolar. This is the difference, the different appearance, because this is in microgram, micromolar gram, and this is the micromolar. This is the other, value. But someone, the expert, using this point, but the other using this point. This is make the audience can confuse, but we concern with one of two this appearance. But some experience will show to the appearance. Yeah. And the other 3.59 microgram in millimeter, or 8.78 micromolar. This is inhibit the activity of hepatocellular PLC5 cell line. This is the good value for anti-cancer. If in the next slide, I will show you about the value of the anti-cancer. The NCI USA criteria, that is less than 20 micromolar, we call it is anti-cancer. If we look at this slide, this is probable curcusin B and jatropon for the anti-cancer. Okay, this is the discussion, the study to analyze curcusin B as anti-cancer agent, and this is the value of the anti-cancer using the cholangiocarcinoma with IC50 is like 4.7 micromolar, And in chronic myelogenic leukemia and human non-small cell lung carcinoma cell line, this is the value, 6.1 microgram per milli and 15 microgram per milli. And we get, this is the comparison about the IC50 for the active confound. And the other, if we get from Sahidin, we get the IC50 is 7.2 micromolar, 10 micromolar, and 5.7 micromolar. Yeah. This is the comparative of cell line if you using Kurkosun B in the other cell line cancer. And the other, we look at and compare that jatropon and glucosin B has also been studied. This is in some cell line, it's like cervical cancer line, or we call HeLa, and colon cancer cell line, or WEDRI. This is the IC50, it's like this. Yeah. I think it's good value, yeah, because this is less than 20 micromolar. And our study in Jatropon in HepG2 cell line in 186 cell line, we get, this is the value, 3.2 micromolar. This is very good value. And we compare with the other standard or gold standard for anti-cancer. This is have the pseudopovir or ribavirin. We know this is the value, it's very high. Yeah. And the other gold standard is sorafenib. Sorafenib is anti-cancer, good anti-cancer, and some using anti-cancer in Indonesia with the specific cancer. This is we compare. This is the point. Yeah, 9.63 and 3.96 micromolar. As the result, this is the cytotoxic of jatropon and glucosin B have a better effect. Again, have hepatocellular calcified cell line when compared to the human cell line. This is good news for our country because we can isolate it jatropon and glucosin B from jatropokurkas. Okay, is the still discussion the mechanism of glucosin and jatropon inhibition growth cells. This is the molecular mechanism has not been clear, but I will show you about the diagram about the my research. I developed this research from 2010 until now, and we get, this is the graphic. The first time I developed this graphic from chlorogenic acid, from Kopi Robusta, from Lampung, and we isolated about it till maybe more than 1 year. And then I test chlorogenic acid in hepatocellular carcinoma in one type, that is HepG2 1886, and this is my pattern, and I have pattern in DT. And then this is my novelty. How about the glucosin B and jatropon? I think this is the same with this mechanism. That is, there are 4 indicators. The first is microarena 146A. is this is the microarena, and I think this is a new gene, and the world developed it about maybe in 2010, and I am claim that is the first time in Indonesia develop about the microarena, and this is my research and this is my novelties. MicroRNA and then cyclin D1 and caspase-3. I treated I treated the chlorogenic acid on hepatocellular carcinoma, or we call HG21886, and then this is the value of microRNA is decreased, and the same with cyclin D1. Cyclin D1 is associated with the cancer. This is the same. If we treated chlorogenic acid, the cyclin D1 is decreased, but different in caspase-3. In caspase-3 is increased. Uh, in the other research, in the other paper, I write and I publish about the microarena and cyclin D1 in the same with the active conform with jatropon and glucosan B. And this is my research. That is microRNA is decreased, and cyclin D1 is increased. Cyclin D1 and microRNA is associated with the activity of the cancer. Okay, the conclusion, jatropon and glucosan B have some potential as anti-cancer agent. It has better cytotoxic activity on the hepatocellular carcinoma, especially in PLC5 type cell line, if we compare with the another human cancer cell line. In addition, jatropon and glucosan B also have better anti-cancer activity on cell line compare if we compare with anti-drug such as sorafenib, sildenafil, rabifilin, which has used widely. Okay, I think this is my presentation. I think 20 minutes, and then I back to Selvi. Thank you, Selvi.
Thank you, Dr. Asep. That was really amazing talks about the natural compound in Jatropha as anti-proliferative agent for hepatocellular carcinoma. That's really interesting that those plants actually have some potency as anti-cancer agent and even have better cytotoxic activity compared to another anti-cancer drugs. Okay, now I'll open up for the discussion session. There is a participant who raised her hand. The name is Evi Nur. Okay, for the host, could you please help me to unmute the microphone for the Evi Nur? Hello. Okay, sorry, maybe we will go to the next question. This is from there's no name of the participants here, but the question is why the mechanism of jatropon and cortisol B on inhibition of cancer cell growth by the molecular mechanism is still not clear. Okay, for Dr. Asep, please, could you please give us the response?
Okay, thank you, Selvi. Yes, that is not clear until now because some experts have experience and trial about it. And then I say before that is our research from 2010 until now. This is I say about the expert is have divide to two way. The first mechanism that is like me. We use the novelty or we use the uh, molecular mechanism with nuclear factor kappa beta. We call it in this slide. Nuclear kappa beta is the major pathway in molecular mechanism. We use it about for there is the indicator. If we treat it with the chlorogenic acid, jatropon, and glucosan B, nuclear kappa beta is down or is decreased. That is my research with we associate with the microRNA 146A and cyclin B1. When we treated it with uh, high dose in chlorogenic acid, my dose for HG21886, we use it about the 700 micromolar. It is very, very high dose. And if we treat it with jatropon and glucosan B, it's like that. Maybe more less than 20 micromolar. This is the indicator that is chlorogenic acid, glucosan B, and jatropon has activity confirmed for the uh, cancer cell line. Yes, that is not clear until now. If you read about the journal international in the world, in the internet, making you confused. But for this dissertation, I think we can get one way, and we should uh, make sure about the this research. Uh, I say to you that is not clear until now. Maybe some researchers have the other mechanism for it. But for now, we need for anti-cancer. This cancer is still grow, and what will we do for this patient? Okay, that is maybe for our explanation about the mechanism for the jatropon and glucosan B therapy.
Thank you, Dr. Asep. Yes, I agree that I think to find the exact pathway or molecular mechanism is still a hard work for us and need a long research to find the exact pathway because my previous research also work on the pathway and it cannot find just by one research only. So it is our work to find the pathway. Okay. Thank you. And then we go to the next question. This is from Muhammad Iqbal. I think this is one of our lecturer. What functional group of jatropon and corpuscle B that responsible for anti-cancer agents? Okay, thank you. Dr. Asep, please give us
Okay,
this.
I will tell you about the cell cycle in jatropon or glucosan B. That's right. Glucosan B and jatropon inhibit the growing about the cycle cell. And if we show you about the cycle cell, that is some gene or some pathway, and that is we inhibited about the growing of the cell. is like the first when the kurkosundi uh, uh, we call it in ligand or in receptor that is can interface to the into inside cell with the kinase C, kinase C, and that is have some cascade until go to the cell. And in the cell, inside in the cell, that is can inhibit cyclin D. Cyclin D1, that is one of the enzyme or gene in the cell that is important for the growing of the cell. When the curcumin B enter in the inside the cell by the kinase C and then inhibit for production of cyclin D1. If this enzyme or this gene inhibit, there is growing of the cell at inhibit 2. And then until one time is uh, with the dose, the cell is cannot growing. And then until is we call is like uh, I forget about the name in caspase 3 that is mitosis not mitosis it's like before cell death I forget about it and then until in the end the cell is dead. Yeah. That is the cascade of how cell can death in the uh, in this research. Okay, Selfie, that is some question about microRNA.
Okay, thank you, Dr. Asep. I think there is the next question also from Muhammad Iqbal, and Muhammad Iqbal already here with us. So maybe you can for Muhammad Iqbal, maybe you can ask your question directly. You can open up your microphone to ask directly to Dr. Asep, please. Because I think this is a really interesting discussion. So maybe you can ask directly. Oh, okay. I will read the question for Dr. Asep again from Muhammad Iqbal. In your prediction, what phase in cell cycle that jatropon and curcumin can inhibit the cancer process based on the predicted mechanism?
Okay, that is I forget about the cycle cell. That is sometime in cycle cell. That is G G G0 until G1 and then G2 and then until the synthesis and mitosis. This is the point of how can the anti-cancer inhibit the growing of the cell. That is, jatropon can, I repeat again, can inhibit the growing of the cell. If the growing of the cell is inference or in terganggu apa ya istilahnya, that is mixed cell is not growing well, and then in the end the cell is can dead. I think this is my explanation. Selfie, maybe your question is can I answer or no?
Okay. Okay, Dr. Asep. I think to talk about cell cycle is also need further research yeah, to find where's the exact cell cycle that inhibited by the cortisol. Like that. And then there's also the next question, Dr. Asep. This is the participant also mentioned his or her name, but the question is he want to ask something that is shown in the presentation about microRNA. The question, can viruses use cellular microRNA to promote their replication?
Okay, this is a good question. MicroRNA, this is the one gene developed in 2010. And no, in 2002. This gene is very small, maybe more or less for the um, maximum is 20 pair of basa. It's very small, and this is the indicator for growing or death for the cell. We interest with this microRNA because this is the new gene, and then it's very small, and we can detect in small concentration for some disease. Uh, in my research, especially for the microRNA 146A, that is can predict about the hepatocellular carcinoma in the next years or in the future years. That is, we call microRNA as the mapping gene because this is very small. And how about now for the virus? I think virus is the same, is like the other microorganism. Virus have RNA, have DNA, is like cell, but virus uh, is very clever for the mutation. It's like in COVID-19 or the other virus that is very we call it is very uh, apa ya, sangat cepat ya, jadi sangat cepat untuk can mutation. It's the same. I think in virus it is the same. MicroRNA is have in the virus, and I think how we can get the microRNA. That is the method on how we can get the virus microRNA. The first time is like we done about the isolated about RNA and DNA. The first time we get the sample maybe from the jaringan or from the blood or from the hair, and then depend on the sample. In my research, because this is the cell line, I do this research and isolated RNA and make copy and inverse it to microRNA. This is the very expensive experience in the lab because there is some agent we buy from the overseas, it's like from Canada and the other country. And it is I highlight about the virus is the same. We can get the microRNA from the virus, and it is possible to make a replication. Yes. In PCR, I think we can replication about the microRNA. I make it until now. I think I put in the nitrogen 1 - 100. I think this is still good condition. This is my answer, Selvi.
Thank you for the explanation, Dr. Asep. Yes, I think that's also based on my experience about microRNA when I got my postgraduate education. MicroRNA is a really interesting target due to its roles in inhibiting and promoting cancer. So up to now, researchers still have a lot of work to do to reveal many roles of microRNA. So that was really interesting. Okay, we have Dr. Khairunisa Brawi here. Maybe Dr. Nisa want to ask directly to Dr. Asep. Dr. Nisa.
Are you there, Dr. Nisa?
Sorry, I can't unmute your microphone. Okay, maybe we go to the next. There's a lot of question, Dr. Asep. I think that's really interesting talk. Okay, there also one question again from Muhammad Iqbal. There are many parts of protein kinase C receptor. And what is the main target of the PKC structural ligand domain? Okay, that's the question, Dr. Asep.
Okay. Thank you, Dr. Iqbal. This is the good question. Kinase C, that is structure ligand domain. I think this is my research because I don't make evidence about the kinase C. But in my pathway, we looking for the pathway about how can jatropon or glucosan B can enter the cell. That I choose this kinase A because this is the more pathway for the active compound because it's like chlorogenic acid and not only for chlorogenic acid, caffeine, jatropon, glucosun, and hydroxyl and pinostrobin. We tried it on my research. I think this is the pathway for the entry the cell. And there are, yes, I agree with you for many parts of protein kinase, but we make sure about this. I don't know exactly about protein kinase 1, 3, and the other, but this is the major pathway for kinase C receptor. If we make the evidence of or why or where kinase C, I think I will do about the other research because we have limitation about the funding and the other. If you know, my research is spent about the 1.6 billion in my dissertation, and this is my concentration. Maybe in the future, I will do for this research. Maybe we together with you and the other pharmacists because this is the I think we need for the some equipment from the overseas if we can do it, maybe in overseas, maybe we make collaboration with the other university. It's like me in 2012. I do my research in Florence University. There is a good laboratory in there because that is very complete, very complete. It's not like in our campus or Indonesia. In Indonesia, maybe you can do this research in Darmais Hospital and maybe some in Hasan Sadikin and maybe some in ITB and the other because in not complete in one university. Yes, this is my challenge. This is our challenge, Serbi. Okay.
Yes, good answer. Thank you. I agree that we need further research. Maybe many next research will have to find the exact uh, answer for this question. Okay, there is already have a question here from Hari Nugroho Eko Suryantono. Dr. Asep, have you ever been checked the peroxide bridge structure of the active compound in jatropon? Maybe it has possibility against cancer. This is from Batam. He is from Batam.
Yeah. Thank you for Hari Nugroho. That is peroxidobrid. Yeah, peroxidobrid. I think maybe we can do for the other research, yeah, for the peroxidase, maybe for DPPH, yeah, maybe we can do in the other research because before I do the peroxidase research with chlorogenic bath for jatropon and Kurkosen B. That is, I don't do this research in peroxide. Maybe that is maybe the structure compound active jatropon maybe can do with this research. For this research, I think I don't do this research on peroxide because I think I do I think enough for do the chlorogenic acid compound and caffeine in the DPPH or peroxide acid. Okay, that is my explanation.
Thank you, Dr. Asep. Yes, again, we need further research maybe to answer this question. Okay, there's already we still have time. There is one question from our students, Dr. Asep. Could I ask about is there any side effect of jatropon and glucosan B? And is this medicine already used in public needs? Thank you.
Okay, thank you for the student. My research in vitro, not in clinical trial. I will tell you about the step by step for the how can the drug used for the human. The first we do in the animal and and then we use in the human. In animal we can looking for the toxicity. We can look at it for the safety, and then after we sure about the activity and toxicity and safety, and then we do in the human. In the human, we call it clinical trial. It's like in vaccine for now. That is have four steps for clinical trial. In the first step in the small population, and then we do in the uh, second phase or second step is more than 100 population, and then we get until the step three or phase three. It's like if vaccine now for the phase three. In phase three, we can elaborate about the some side effect and some maybe that is not on the paper or not on the book. We can get it in the human or on the patient. And after phase three, we go to phase four, or we call it about the post marketing surveillance in the phase four. And I will tell you about effect, side effect in the public. I think it's a long journey, long journey, because we should spend about maybe 100 billion, maybe, and maybe more, because it's a long journey. This my research only in laboratory, and we seek about the factory or the other university to join this research. Before I have present this research in uh, Kentucky University in 2007 when I go to the USA and the Wright State University. But this is not continue about the research. Maybe this is the problem about the money, maybe. And then some active compound, this is very difficult to send to overseas because we have about the rule about it. Okay, I think enough.
Okay, yes, I agree. Again, Dr. Asep, this is still based on laboratory data. So, to be commercial use in public, we still have a long journey. We still need to go through the clinical trials in some phases like that. Okay, Dr. Asep, maybe that was the last question because we are running out of time. Okay, there was a really great talk and discussion from Dr. Asep about Jakarta podcast and its anti-proliferative activity. And now we're coming to the end of this session. I would like to apologize for the person whose question cannot I read one by one due to the time limit. And also on behalf of the conference committee and also to all the attendees, I would like to say thank you for Dr. Asep for the amazing talks. Hope the talks would enrich our insight and knowledge would enrich our insight and knowledge about future. So everyone, distinguished guests and all participants, I would like to conclude this session. I apologize if I'm making mistake in assisting this section. Thank you again for Dr. Asep for your time and your really interesting talk. And I pass back this session back to the host. Thank you. Have a nice day, everyone. And wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Yeah, happy success for you. See you.
Thank you. Yes.
Yeah. Assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you so much for our incredible speakers along as our moderator. We already hear the conclusion of this first lecture from Ms. Selfie.
Okay, and for all the participants, don't forget that there will be a link for you all to fill out in the columns.
Aduh, nanya aja udah lama, udah 5 tahun lebih di mana teorinya.
Okay, sorry. So Once again, for all the participants, don't forget that after this, we will put a link for you all to fill up for your presentations here in the column session after the next sessions for our next invited speaker. So, ladies and gentlemen, we will begin to the next seminar by our invited speakers, such as Professor Dr. Zeli Nurrahman, Dr. Tawi Purangedara, and Dr. Miroslava Rizova.
And it will be moderated by a lecture from Faculty of Medicine, Lampung University, Dr. Hairuni Sabrawi Asket MKes IFO. But beforehand, please let me read the CV. Dr. Hairunisa has been graduated and got her Bachelor of Medicine from Sriwijaya University, followed by taking a master degree in biomedic in University of Pajajaran and doctoral program in University of Andalas. Dr. Hairunisa has published 6 international journal and 3 national journal in the past 5 years and got intellectual property right or HAPKI with the title Obesity and Sport Medicine. Please welcome Dr. Hairunisa.
Assalamualaikum warahmatullahi wabarakatuh. Thank you. Okay. With discussion 20 minutes. Maybe you could make it 30 minutes, Prof. Please, Prof. Time is yours.
Okay, thank you. Good morning. Thank you for organizing committee that give me opportunity to give a talk and international conference. May I share my presentation? Already come up?
Come up, Prof. Proteins of SARS-CoV-2.
Okay. Actually, my background is biochemistry. I'm interested on biopigment from microalgae, but since our institute is closed due to SARS pandemic, so I just turn and then make and read everything published concerning SARS-CoV-2. So in this presentation I'm focused on protein of SARS-CoV-2. SARS-CoV-2 is a single-stranded positive sense RNA virus. On the basis of genomic organization and phylogenetic relationship, SARS-CoV-2 belongs to the genus coronavirus and family Coronaviridae. SARS-CoV-2 has approximately 30 kilobase genome size. The structure of SARS-CoV-2 is found to be similar to SARS-CoV with variant size ranging from 70 to 90 nanometer. Spike, membrane, and envelope surface viral protein of coronavirus are embedded in host membrane lipid bilayer. The the helical nucleocapsid protein covers viral protein. Spike glycoproteins on the surface SARS-CoV-2 mediate interaction with cell surface receptor ACE2, angiotensin converting enzyme 2, is to provide the entry point for the coronavirus to hook into and infect the whiteness of human cell. The size of coronavirus genome is in the range of 29 to 32 kilobase. This is available on GISAID database. There are no significant difference among SARS-CoV-2 and SARS-CoV. SARS-CoV-2 genome has 11 open reading frames, with two flanking untranslated region at 5 VN of 265 and 3 VN of 378 nucleotides. The genome of SARS-CoV-2 lacks the hemagglutinin esterase gene. The RFF encodes 9,680 amino acid polyprotein. The first offering ORF comprise approximately 60% of genome encodes certain non-structural protein (NSP), whereas the remaining open reading frame encode for the accessories and structural protein. Spike protein, envelope protein, membrane protein, nucleocapsid protein, 3CL protease, papain-like protease, RNA polymerase, and helicase protein have been suggested to be viable for anti-drug targets. The structure of spike glycoprotein structure of spike glycoprotein of SARS-CoV-2 resembles that of the spike protein of SARS-CoV. Like SARS-CoV, SARS-CoV-2 use the ACE2 receptor for internalization and TPRSS2 serine protease for S protein priming. The genome of SARS-CoV-2 is similar to genome of coronavirus that caused SARS epidemic in 2003. Much of understanding of protein found in SARS-CoV-2 are based on the numerous research studies report on SARS-CoV and other related viruses like MERS-CoV. SARS-CoV-2 has a total 11 genes with 11 ORF, ORF1, 1AB, ORF2, or spike protein, ORF3A, ORF4, envelope protein, ORF5 for membrane protein, ORF6, ORF7A, ORF7B, ORF8, ORF9, and ORF10. The protein of SARS-CoV-2 consists of two large polyprotein, ORF1A and ORF1B, that proteolytically cleave from to form 16 non-structural protein, 4 structural protein like spike, envelope, membrane, and nucleocapsid, and 8 accessories protein. Similar to SARS-CoV-2 lacks the hemagglutinin esterase gene, which is found in human coronavirus. SARS-CoV-2 is thought to have originated from its close relative, bad cough rat G13, which is isolated from horse bats. SARS-CoV NSP1 induced a near complete shutdown of host protein translation by two strategies. First, NS1 bind the small ribosomal unit and stall conical mRNA translation at various stages during initiation. Second, NSP1 binding to the ribosome lead to the endonucleolytic cleavage and subsequent degradation of host mRNA. NS1 inhibits all cellular antiviral defense mechanisms that depend on the expression of host factor, including the interferon responses. This shutdown of the key parts of innate immune system may facilitate efficient viral replication and immune evasion. NSP1 of SARS-CoV-2 show 84% amino acid sequence identity with SARS-CoV, such as things similar properties and biological function. The C-terminal residue lysine 164 and histidine in 165 in SARS-CoV are conserved in beta coronavirus and essential for the S interaction. S mutation to alanine abolish for the S binding and relieve translational inhibition. A loss of NSP1 function may render the virus vulnerable toward immune clearance. Its central role in weakening the antiviral immune response, make NSP1 a potential therapeutic target. NSP1, NSP2, and NSP3 are processed by one or two papain-like proteinase. Polyprotease, cleavage, and terminus of a replicase polyprotein to release NSP1, NSP2, and NSP3. This step is essential for viral replication. NSP2 is a RNA binding protein that accumulates in cytoplasmic inclusion. NSP2 is involved in coronavirus genome replication. NSP2, along with NSP1, are involved in suppressing gene expression. NSP3 has almost viability in the protein domain from one group to another. It's believed that NSP2 and NSP1 protein evolve with NSP3. NSP1, 2, and 3 may mediate host-specific function depending on coronavirus group. NSP4, this is a little information on this protein. This protein interacts with NSP3 and possibly host protein to confer a role related to membrane rearrangement in SARS-CoV. Moreover, the interaction between NSP4 and NSP3 is essential for viral replication. The next, I'm going to talk about NSP5 protein. This is a main protease of SARS-CoV-2, is a key enzyme of coronavirus, and has a pivotal role in mediating viral replication and transcription. 33.8 kilodalton main protease is also known as 3C-like protease or chymotrypsin-like protease. Embro with 306 amino acid residue long is composed of three domains. Domain 1 and domain 2 have anti-parallel beta structure, and domain 3 contain 5 alpha helix arranged into large anti-parallel globular cluster. And it is connected to the domain 2 by a long loop region. SARS-CoV-2 of Embro has cysteine and histidine catalytic diet, and the substrate binding site is located in the cleft between domain 1 and domain 2. Main protease digests the polyprotein at least 11 conserved sites, starting with autolytic cleavage of the enzyme itself from polyprotein B1A and polyprotein 1B. The function important of main protease in the viral life cycle combined with the absence of closely related homologies in human. A half maximum effective concentration, or EC50, of epsilon against SARS-CoV-2 is 4.0 67 micromolar. Meanwhile, using N3, this is peptide-like, is 16.77 micromolar. NSP6 protein of the avian coronavirus has shown to generate autophagosome from the endoplasmic reticulum. Autophagosome facilitates assembly of replicase protein. NSP6 limited autophagosome or lysosome expansion, which don't prevent autophagosome from delivering free viral component for degradation in lysosome. With SARS-CoV-2, NSP6 was shown to induce membrane vesicle. Work with NSP3 and NSP4, NSP6 make virus factory bubble. A complex of NSP6 and NSP7 and NSP8 and NSP12 yield the RNA polymerase activity. NSP8 is a peptide cofactor that makes a heterodimer with NSP6, and this NSP6 and NSP8 heterodimer complex with NSP12. In addition to NSP7 and NSP8 heterodimer, an NSP8 monomer unit also complex with NSP12, which ultimately form RNA polymerase complex or RDRP complex, RNA-dependent RNA polymerase. RDRP of SARS-CoV-2 is composed of a catalytic subunit known as NSP12 as well as two accessory subunits, NSP8 and NSP7. Remdesivir inhibits the RDRP of multiple coronavirus. NSP9 is thought to mediate viral replication, overall virulence, and viral genomic RNA reproduction. NSP9 protein is similarly obligate dimers. NSP9 may associate with unprocessed viral polyprotein, retaining them near the virus RNA. Human cells have antiviral protein that find viral RNA and shred it. NSP10 work with NSP14 and NSP16 to camouflage the virus gene so that they don't get attacked. NSP10 interact with NSP14 in SARS coronavirus, and this interaction stimulates the activity of NSP14. NSP14 is known to function as an adenosyl methionine dependent methyltransferase or MTAs. Furthermore, NSP10 stimulates the activity of NSP16 with a O-methyltransferase. RDRP domain resembles a right hand comprising the finger, palm, and thumb subdomain that are found in all single subunit polymerase. Subunit NSP7, NSP8 bind to the thumb, and additional copy of NSP8 bind to the finger domains. NSP12 subunit bind to the first strand of RNA between its finger and thumb subdomain. The active site is located on the palm. subdomain and is formed by 5 conserved NSP12 elements known as motif A to E. Motif C bind to RNA 35N and contain residue aspartic 760 and 761, which are required for RNA synthesis. The additional NSP12 motif F and G reside in the finger subdomain and position the RNA template. As RNA duplex exits from the RGRP left, it forms the second helical turn that protrude from the NSP12 surface. The protruding exciting RNA duplex is flanked as a long alpha helical extension that form a highly conserved N-terminal region in 2 NSP8 subdomain. Remdesivir function is immediate RNA chain terminator. NSP12 called helicase enzyme unwind duplex RNA. NSP NSP13, NSP13 with hollow RDRP can enhance the helicase activity of NSP13. In addition to its helicase activity, NSP13 of SARS-CoV-2 is also known to boost 5-feed 3-phosphate activity, which is responsible for introducing 5-feed terminal caps of the RNA viral. Both eukaryotic and most viral mRNA have a 5-feed terminal cap structure. These 5 terminal caps in the site of recognition for translation and play a role in splicing, nuclear export, translation, and stability of mRNA. NSP14 with NSP10 is important for viral replication and transcription. The N-terminal exoribonuclease domain play a proofreading role for prevention of lethal mutagenesis, and the C-terminal domain function of NSP14 is for methyltransferase mRNA capping. NSP16 for SARS-CoV-2 is characterized as endoribonuclease that cleave RNA at unoriginate at 3' position to form to 5 and 3' cyclic photodeesterase product. NSP14 specifically target for degrade the viral polyuridine sequence to prevent the host immune sensing system from detecting the virus. The viral RNA has 5 caps which protect it from mRNA degradation by exoribonucleases, promote mRNA translation, and prevent the viral RNA for being recognized by innate immunity mechanism. RNA caps in methylated guanine nucleotide connected to triphosphate bridges to the first transcribed nucleotide. NSP16 methylates to 5-hydroxy group of adenine using S-adenosyl methionine as methyl source. This methylation, methylation indispensable for replication of coronavirus in cell culture. This is a S protein that known. The gene for spike protein in SARS-CoV-2 has an insertion of 12 nucleotides. This mutation may help the spike bind tightly to human cell. This is a crucial step in its evolution from a virus that infected bats to other species. Glycosylated S trimers bind the ACE2 receptor and mediate entry the virion into target cell. In the perfusion form, the receptor binding domain sits at the top of a broad turmeric spike above the fusion core. Three copies of receptor binding domain are surrounded by three copies of N-terminal domain, which show some mobility. In close perfusion conformation, all three copies of ribosome binding domain lay flat on the spike surface, largely including the receptor binding set, while the open perfusion conformation where a multiple ribosome binding domain lift up to expose the receptor binding. The surface of trimer is extensively glycosylated with 22 potential N-leak glycosylation sites per monomer. After receptor binding, structural transition of perfusion to post-perfusion from ring to fusion peptide and the transmembrane domain together at one end of a long needle-like structure centered around a three-helix bundle. 5 N-link glycan are spaced along length of post post fusion post-fusion spike. The infill protein is a small integral membrane protein in a coronavirus, which can oligomerize and create an ion channel. From homology modeling, the most favorable structure could function as kit ion channel conducting proton ion. The infill protein play multiple roles in the viral replication cycle, like viral assembly, virion release, and viral pathogenesis. The SARS-CoV membrane protein is integral membrane protein that play an important role in viral assembly. SARS coronavirus membrane protein so to induce apoptosis. Membrane protein interact with nucleocapsid protein to encapsulate the RNA genome. The nucleocapsid protein of coronavirus is structural protein that bind directly to viral RNA and providing stability. The N protein of SARS-CoV-2 has been found to antagonize antiviral RNAi. In other studies, the nucleocapsid protein of SARS-CoV-2 inhibit the activity of cyclin-dependent kinase, cyclin CDK complex. After infection, the N protein enters the host cell together with the viral RNA to facilitate its replication and process the virus particle assembly and release. SARS-CoV-2 N protein contain two distinct RNA binding domain, the N-terminal domain and the C-terminal domain linked by a fully structured linked region containing a serine arginine-rich domain. Due to the positive amino acid SARS-CoV-2 N-terminal domain and C-terminal domain have been reported bind with viral genome. So from the protein that we can found in SARS-CoV-2, so we can obtain SARS-CoV drug by designing antiviral compound and then test it based on understanding of structure and function of SARS-CoV-2. Okay, okay, thank you for your attention.
Thank you for Prof. Jelly. It's so amazing presentation. Try to explain to for all us, for all participants, that how complicated SARS-CoV-2. But that's I heard that something interesting, that there is not big difference between SARS-CoV-2 and other SARS-CoV. Maybe you could explain first, Prof, for this about there why you say that there's no difference between SARS-CoV-2 and other SARS-CoV before we
Yeah,
continue with question, Prof.
okay, thank you. Because this is a high homology identity on the SARS-CoV-2 phones. So based on the high similar homology, we can explore that protein SARS-CoV-2 is a group of SARS-CoV. So everything information that we found already in SARS-CoV may can be used to explain the function of SARS-CoV-2 protein. So this
So,
based on high homology or
so
high identity on of sequence.
okay, so Prof, when you say that there's no difference, when you explain about this containing of this SARS-CoV-2 protein, like 67% genes for 10 non-structural protein and 33% for structural protein, it would be similar with other SARS-CoV. It's just like that, or maybe the difference is more deep than that. Okay, Prof. Maybe we'll continue with the question, the answer and question, if you don't mind, before we
Yeah,
waiting for other Mrs. Miroslava Risova. Excuse me. How if we continue for question, or if you don't mind, how you agreement for this, Prof? We could make in pleno, or we could make every session. How it could not, if we work in plenary discussion, right? So maybe before continue with question and answer, maybe better if Prof. Jelly and other and me listen about the presentation of Mrs. Miroslava Risova, because she's already here. We appreciate for this. Thank you, Miss Miroslava Risova from Czech Republic. Okay, before we continue with the second presentation from our second speaker, invited speaker, permit me to reading the curriculum vitae from this second speaker, the Honorable Miss Ing Miroslava Risova from Czech Republic. His economic education and academic degrees on 2012 as the 12 as doctoral degree, PhD, in applied science in engineering nanomaterials, Faculty of Mechatronic and Interdisciplinary Studies, Technical University of Liberec. Oh, sorry. Okay, I continue. Mrs. Miroslava Risova, excuse me. There's about something to arrange about the rundown. Maybe I permit to before we'll continue question and answer and presentation of you and Professor Jelly. Maybe I'll back first to Mr. Moderator, and sorry, Prof. Maybe we will arrange. We will change the arrange of the rundown. Sorry, maybe I'll back to the moderator first. Professor Jelly, I hope you could waiting for the session because we have to declare to notification about a few information about this symposium. Sorry, Prof. and Miss Miroslava. I hope you both could waiting for this. Thank you. Okay, Mr. Moderator, I'll back to you. The discussion will continue after the notification. Okay, thank you.
Thank you so much for our incredible first invited speaker as well as our moderator.
Okay. Next, we have announcement for the three best presenter oral presentation yesterday. So for the announcement, we will announce you from the first best presenter for the oral presentations. Selvi Rahmawati from Universitas Indonesia for Universitas Lampung with total score 131. And second, Dian Widianti from Universitas Yarsi with score 130.5. And third, Nur Ahmad Rudin from Universitas Gajah Mada with total score 129. Okay. So I'm gonna announce once more time about the three best presenters for the oral presentation yesterday. For the first winner is Selvi Rahmawati from Universitas Lampung with total score 131. And the second winner, Dian Widianti from Universitas Yarsi with the score 130.5. And the third winner, Nur Ahmad Rudin from Universitas Gajah Mada with total score 129. Once more, congratulations for all the best presenters in the oral presentations.
Once again, congratulations for all the three winners of the best speakers in oral presentations. So the next one, we will show you the video of ICAM's 2020. Please enjoy.
Thank you for your nice attention. So next, we're gonna move again to the next sessions with others invited speakers. So we'll be back with the moderator, Dr. Nisa Khairunisa Barawi. Please welcome.
Thank you, Mr. Moderator, Taufik, my student. Thank you for Professor Jelly and Dr. Miss Miroslava Ristova for writing this, and thank you for the understanding situation. Okay, maybe we'll continue with second speaker, invited speaker, Miss Miroslava Ristova, and before that, I'll continue to read her curriculum vitae that's named in Miroslava Ristova from Czechia Republic, Education and Academic Degree 2012, recent doctoral degree, PhD, in Applied Science in Engineering, Nanomaterials, Faculty of Mechatronics and Interdisciplinary Studies, Technical University of Liberec, correlate with this presentation today about nanotechnology, and 2012 Magister Science degree in Material Engineering Textile Faculty, Technical University of Liberec, Diploma with Honors. 2010 BA degree, Bachelor degree in Design, Textile Faculty, Technical University of Liberec, Diploma with Honors. 2008 Bachelor SC degree in Textile Materials and Technology, Textile Faculty, Technical University of Liberec, Diploma with Honors. Okay, and he has professional profile as researcher PhD student, supervisor of the Biomaterial Processing and Cell Culture Lab of the Department of Applied Biology at the Institute for Nanomaterials, Novel Technologies and Innovation, Technical University of Liberec, Czechia Republic. Her research interest about biomaterial-based structure and many research correlate with nanotechnology, and I so interested with this because this is similar update with COVID information from Prof. Jelly, and teaching experience and supervised thesis teaching nanomaterial application in medicine, safety and legislation of material, because we know there is contradiction about nanotechnology for medical worker that few scientists suspect that there's harmful for worker, but maybe Mrs. Maslova could explain this. Okay, she has many recent publication. Maybe I could say in for 2020 he has was water treatment by novel polyamide polyethylene nanofiber with immobilized lacase 2020, and few others just like in 2000, just like cyclohexane 1.2 diamine based organosilane fibers as a powerful tool against pathogenic bacteria. It's so interesting, and many publication just like Professor Jelly. And now he is participant in project research in 2020 about biologically active materials for application and medical devices, nanomedical technology, University of Liberec, and principal investigator for TUL, 2020 until from 2019, technology for high throughput production of drug delivery system in Occur Technical University of Liberec as principal investigator for TUL. Okay, and for recent two, Science 2018, hybrid materials for hierarchical structures, Technical University of Liberec as senior researcher, supervisor of activity, and she's a pattern and industrial collaboration about three, just like commercially used pattern and utility models, correlate with nanotechnology, one cover with nanofiber layer for drug delivery, multifunctional self-standing diamond porous supports for cells cultivation, and nanofiber structure with increased surface functionality and immobilized organic agent. For pattern and industrial collaboration, I could not to read because it is so important for researchers just like me, just like us. He has membership of the European Society of Biomaterial and other experience. Okay, there's a bit curriculum vitae from our amazing second speaker, Ing Miroslava Risova. Time is yours.
Thank you very much. I would like to say it's a very big pleasure for me to be here and be allowed to give this talk as invited speaker. Thank you very much. If I understand it, you will be sending the slides, right, with the presentation. As it was kindly mentioned, my work has been focused on application of nanomaterials, mostly in biomedical field, but also in water treatment. So today I would like to focus on some research experiences we have with biomaterial-based nanofibers, their applicability in a medical field, and their advantages and also some limitations in the application. Shortly, I would like to mention a few words about the Technical University of Liberec. So next slide, please. As you probably know, Czech Republic is located in the central of Europe, and our city and our university is located right on the north, very close to the borders with Germany and Poland. So most of our research is kind of international collaboration based. We are very proud to be called a place where the nanospider device was born. And that's mostly the reason we are working so extensively with nanomaterials, mostly nanofibers, because it has very huge tradition in Technical University of Liberec. Next, please. The Technical University of Liberec has over 8,000 students up to the date. It consists of 7 faculties and a newly built institution, which is called Institute for Nanomaterials, Advanced Technologies and Innovation, which is exactly the research institute I'm based. It was set in 2012, and its main goal is cooperation with industry and linking the basic research usually realized at the university to the industry and translation of the research obtained. So we are very proud with the tens of patents we've been already able to process, and many collaborations with not only companies in Czech Republic, but also around all the world. Next, please. So as I mentioned, Technical University is called the birthplace of nanospider device. Many of you might be familiar with the electrospinning technology. The principle is rather simple. It's based on application of high voltage onto polymer solution or melt, and transaction of the polymer mass into the collector, which is usually grounded or oppositely charged. There are several stages of the electrospinning devices, giving it the applicability according to the amount of nanofibers you are able to obtain. The needle electrospinning is the very basic one, but as application of this technology approach around the world, there were many new technologies developed in order to induce higher production of the technology. So that led to development of needleless electrospinning, which is basically the principle of the nanospider device, which was developed and patented at the Technical University of Liberec in 2005 by a team of Professor Irsa. Some schematic pictures of the nanospider device and its principle you can see on the left side of the screen. The principle is very simple, as I mentioned. The first generation of the device was based on rotating drum covered with a polymer solution, creating hundreds and thousands of Taylor cones, and which are used as a source for the polymer solution, forming the nanofibers during the process. Usually the nanofibers are placed on the support in form of textile. It can be microfibrous textile, it can be yarn or any other structure which is suitable for your application. Huge advantage of this device is homogeneity of the nanofibrous layer through all the structure or the material, which is usually wide 50 centimeters, and you can create in a continuous process several hundreds or thousand meters in one row. So you can see that the capacity and production can be very high and is designed directly for the industrial production. Next, please. Thank you. Over the last 2 or 3 decades, many polymers were electrospun for not only medical but also technical applications. Usually it's said to be over 200 different polymers and polymers mixtures. Among these, the biopolymer play very important role, because when you design material for tissue engineering, drug delivery, or some other medical application, you are very interested in its functionality and also, of course, its biocompatibility, degradation. So these are all the advantages that biopolymer can give you when it's processed through electrospinning and the nanofibers are formed. Among the advantages like biocompatibility and biodegradation is also variety of functionality, possible bioactive effects depending on the polymer of your choice. And very important thing is sustainability, because in contrary to synthetic polymers, it's possible to use some of the base source obtained materials. Usually this is the case of chitosan, for example, or silk fibrin. Of course, there are usually some disadvantages for each of the material. It's important to mention that with the biopolymer coming from natural sources, it can be very problematic to keep its stable quality because of possible environment polluted impact. And of course, each electrospinning material and each electrospinning solution is very unique, mostly from the solvent system point of view. So when you are preparing material for medical applications, it's very crucial to choose only biocompatible or medical approved solvents, which is not always the possibility. So not all the biopolymers you would like to are basically able to be spun to the nanofibers and used in a medical application with approval of the authorities. Next, please. So in my lecture, I would just like to shortly compare 4 of the main types of the biopolymer-based nanofibers we've been working on for last, let's say, 5 or 6 years. Each of them has very unique properties and different field of application and medicine based on its properties. First of the nanofibers we are working with are the silicon dioxide nanofibers. They can't be really considered biopolymer because it's not coming from the natural source. The precursor is synthesized through the wet chemical synthesis way. But the outcome, the final fibers, are fully biocompatible and biodegradable and possess some very unique properties. So the synthesis, long story short, the synthesis of the nanofibers is formed or is done from the TIOS, tetraethyl orthosilicate as precursor through Stauber salt gel method. When you obtain polymer solution with gradually going polymerization reaction, which leads to formation gel, xerogel, and at the end you can obtain the nanofibers you want. It's very crucial in which part, in which time of the production you involve the electrospinning process, because if you wait too long, you just obtain the xerogel and 3D structures, and it's not possible to electrospin the outcome solve. It's very important that one of the big advantages of the silicon dioxide nanofibers is that it's really possible to obtain different structures, different thickness of the nanofibers, which gives you a different degree of the porosity and of course the surface available for example, conjugation of biomolecules also strongly depends on it. So as you can see on the slide, there are structures going from fiber diameter around 100 nanometers, which could be called like proper nanostructure, up to submicron fibers. But it's important to mention that with these change in fiber diameters, not only surface area is changed, but also, for example, the mechanical properties of the nanofibers, which is also very important property for application. Next, please. One of the very important reasons for application of silicon dioxide nanofibers in a medical field is their unique way of degradation. You might probably seen some images of common biopolymer-based nanofibers and their degradation. Usually, the procedure of the degradation involves swelling of the nanofibers and then slow dissolution or disruption through enzymes in the tissues. With the silicon dioxide, even if you speak of nanofibers or about nanoparticles, which are very commonly used nowadays, you can see very different procedure of degradation, which goes from the inner core to the outside surface. So you can see that from the very smooth nanofibers, within a couple of days, you obtain just a fragile structure, and the surface erosion is quite nicely seen after even 24 hours. The quick degradation of these nanofibers can be very beneficial, actually, and that's why these nanofibers are very perspective for application in wound healing, for example, or tissue regeneration. The reason is very simple, because one of the main products of the degradation of these fibers is orthosilicic acid, which is very beneficial for wound healing and cell proliferation. Of course, depending on the concentration, just like every drug. But it was proven by several papers that orthosilicic acid can strongly affect the regeneration process when it's released into the bloodstream or into the tissue. Next, please. So here are just some results we obtained by testing of our silicon dioxide nanofibers. Of course, before every transduction of the nanomaterial to the market, if you really want to apply it in the medical field, it's very crucial to go through all the necessary, all the obligatory steps for biomedical testing, involving in vitro, ex vivo, and many others, depending on the tissue of interest. Especially, well, actually, this is the legislation in the Europe. So with the silicon dioxide nanofibers, it was proven for them to be biocompatible, non-skin irritant. Actually, as you can see, there is, thanks to the released orthosilicic acid, good impact in increasing viability of the skin cell. And what is very important to mention is that they are non-mutagenic in any concentration we tested, which was confirmed by the chromosomal aberration test. Next, please. And due to their quick degradation, their application for wound healing, for example, and drug release in the site of wound is very obvious. But it's important to mention that there are also some other advantages. Mostly, the quick degradation limits the adhesion of cells to the nanofibers, which means that, for example, in application in wound healing, when you treat infected wounds with these nanofibers, they don't tend to adhere to the wound, to the bed on the wound, which is very crucial, because with some other non-degradable nanofibers which were tested for this application, the main issue and disadvantage was inability to get the nanofibers of the wound after 72 hours, because they were fully adhered to the wound, and the procedure was mostly painful for the patients. So this is not the case of the silicon dioxide nanofibers, because as you can see on the upper row, even though the cells are trying to colonize the fibers by their focal adhesion, they are unable to the surface corrosion and the degradation of single nanofibers. This effect actually is beneficial not only for not adherence to the wound bed, but also it limits adhesion of bacterial strains to the surface of nanofibers. Which you can see on the lower row, where we tested adhesion and viability of two types of bacteria, Staphylococcus aureus and Pseudomonas aeruginosa. As you see, the results were definitely better with the Pseudomonas strain, where not only lower number of bacteria were adhered to the nanofibers, but also after 1 hour exposure to nanofibers, most of them were dead because they had no surface to adhere. Next, please. Regarding the silicon dioxide nanofibers, I mentioned their huge applicability for drug delivery and wound healing mostly. One of their main advantages is also their surface, which can be functionalized with grafting of different functional groups, which can be afterwards used for conjugation of different drugs or biomolecules. In our experiments, we already worked with conjugation of antibiotics and septics, or with conjugation with enzymes for wound debridement. Both of these experiments worked very well, actually. And for example, in case of the enzyme conjugation, it was proven that the covalently conjugated enzyme to the nanostructure is several times more stable, and the enzymatic activity is prolonged by the application in the conjugated state. So just to mention, by the process of silanization, usually or mostly the groups like primary amino, thiol, or epoxy, afterwards changing into the carboxyl group, can be used for further conjugation of the choice. Next, please. Yes, here are some schemes of the process of the conjugation and further conjugation of the biomolecules. It's possible to conjugate. For example, we worked with polysaccharides and the enzyme mentioned. The huge advantage is already mentioned high available surface of the nanofibers, which usually leads to extensive amount of molecules homogeneously binded to the surface of the nanofibers. As you can see on the SEM micrograph, after the conjugation, the fiber structure remains. It's only covered with the polymer on each single fiber. So it doesn't basically impact the porosity of the structure, which is very important in wound healing application. Because it's important to retain the oxygen change with the environment around the wound to prevent any further bacterial infection and its support. Next, please. Here I would like to just show some of the results we obtained and are ready to be published, actually. This work was focused on conjugation of antibiotics on the surface of the silica nanofibers. So the procedure was realized the way I mentioned before. The surface of the nanofibers was silanized, and afterwards the antibiotic was conjugated to the surface. The antibacterial activity was tested and confirmed on two different bacterial strains, E. coli and Staphylococcus gallinarum, and it was proven to have sustained release for 72 hours, which is quite beneficial because within the medical practice, it's very difficult to keep changing the active wound dressing more often than once in 2 or 3 days. So this results proven the real applicability of these fibers and this concept in the treatment of infected wounds or ulcers. Actually, nowadays, ulcer treatment and the wound treatment related to diabetes is a very strong topic because, as you probably know, the numbers of patients with diabetes and related complications grow every single year, mostly in the Western world. So it's expected that this treatment could have could play an important role in this field. It's important to mention that this technique doesn't intend to replace the intravenously conjugated drug or adverse drug, because usually with the wound infections, the infection is spreading not only through the wound but also through all the body. So it's necessary to support this therapy by intravenously or perorally given drugs or antiseptics or antibiotics. Next, please. Thank you. In comparison to the silicon dioxide nanofibers, I would like to talk now about chitosan nanofibers. This is a very good example of the natural source material, which has huge application mostly in medicine, but also in different industrial fields. As you probably know, there are many different sources, including crustaceans or fungi around the world, which are used for obtaining chitosan and subsequently chitin and subsequently chitosan by the deacetylation process. It's very important to mention that this polymer is not only biocompatible, but it's also proven to be biogenic, to possess some very specific bioactive properties, which are mostly given by its polyionic nature. One of the disadvantages can be the already mentioned instability of the source. We've been working during our expertise with 7 different chitosans from different suppliers, and actually this is what we saw mostly. Well, it was very different or very difficult to obtain the same results with different batches from the same producer, which is very crucial for electrospinning method, actually, to have the same quality of the material every time. Next, please. Here are again some morphologies of the nanofibers that you can obtain from the chitosan electrospinning. It's very important to mention that for the electrospinning process, the deacetylation degree of chitosan should be at least 50%. Otherwise, the electrospinning process is very unstable. The morphologies that can be obtained can range from true nanofibers with diameter below 100 nanometers up to microfibers or mixture of microfibers and nanofibers. Actually, a mixture of these two types, micro and nanofibers, were proven to be very interesting material for tissue engineering of various tissues, because it's strongly resembled by cells, and it gives very quick colonization and good motility through cells through the material itself. Next, please. With the chitosan and nanofibers, it's very important to mention that it's polyionic. To be precise, it's polycation. It's a strongly charged nature, gives the chitosan bioactivity and antimicrobial activity against several bacterial strains. As you can see on the picture in the center of the slide, chitosan itself has been proven to give very good antibacterial activity against Staphylococcus gallinarum. This is tested through formation of inhibition zone around the sample. So the diffusion zone was formed in the distance of several millimeters around the tested nanofibers. Of course, with the application of medicine, this can be a very good point, which actually needs to be supported with addition of some other molecule to obtain proper antibacterial effect. The huge advantage of chitosan is that it can be really combined with drugs of basically any type. It can be easily combined with antibiotics or antiseptics, natural compounds, or even antibacterial ions, because it has natural
Excuse
way
me,
to.
Mrs. Miroslava,
Yes.
your time only around 1 minute, maybe.
Oh,
Yeah.
oh my God,
Because
sorry.
this is you still has two nanofiber. There is
Oh, yes.
fibroin and collagen nanofiber. Maybe you could make it faster.
Quicker. Yes, yes, of course.
Excuse
I'm
me,
sorry.
but
No,
it's
no, no.
interesting. Can't
Okay.
finish. Yes, okay.
Okay, I will be much quicker. So I only wanted to mention that it's possible to conjugate also, for example, silver or zinc ions or nanoparticles to the nanofibers and form strongly antibacterial composite. Next, please. Uh, actually, the chitosan can be used in wound treatment, and one very important property of it is also its muco-adhesive nature. So this work has been patented at the Technical University of Liberec, and it's the nanofibrous patches, muco-adhesive nanofibrous patches for sublingual vaccination, which is based directly on its ability to adhere to the layer of mucus below your tongue. Next, please. I would like to also mention silk fibroin as one of the very interesting protein materials which can be electrospun quite easily. It's very popular material because it comes from the waste material the waste material is used for the industrial applications. Actually, with the medical applications, you need to use primary source, but a huge advantage is ability to obtain not only nanofibers, but also 3D sponges and so on. Next, please. They have very different degradation. So basically, this is the process to obtain the silk fibroin through the degumization process and getting rid of the sericin of the surface. Next, please. And here are the structures. The huge advantage of the silk fibroin structures is that you can design the degradation kinetics through the way of the cross-linking you use. Usually it's the ethanolic dehydration, but there are many other ways of the cross-linking. Next, please. And this material can be applied not only in tissue regeneration and wound healing, but also in the tissue engineering of bones with the strong cross-linking. It's non-degradable for 3 months at least. Next, please. Yes,
Maybe you could continue with the explanation only for collagen
Yeah, okay.
nanofibers, because it is so interest once more, because I want to know about nanofibers technology much more today, because it's so update for medical treatment, medical approach.
Okay. Yeah.
Next, please.
Yes. Next, please.
Yeah. Collagen nanofiber.
Yes,
Last.
collagen. I only wanted to say that with the cell it has a huge potential in bone tissue engineering and also in burns treatment because it's non-irritant to the burns patient. Very important biopolymer which can be electrospun also is collagen, which is strongly bioactive. As you can see on the surface, on the top lane, there is comparison of histological analysis of soft tissue, and it's compared to the SEM pictures of the collagen-based nanofibers. You can see the strong resemblance in the structure. Actually, this is the reason why it's mentioned that the collagen nanofibers or nanofibers in general are very potent in tissue engineering because there's strong resemblance between the natural structures in your body and the electrospun nanofibers, and cells can easily recognize this. We tested the collagen fibers not only for soft tissue, but also for bone tissue, tissue engineering, and it led to increase in osteocalcin and alkalic phosphatase production in bone cells, in osteoblasts, which is great confirmation of the applicability for this field. Yes.
Continues.
Actually, yeah, this is the last slide.
Last slide, okay.
Thank you so much. Maybe if you could show the last slide, because there is just the list of my colleagues. Of course, this is not only my work, it's a teamwork. So I would like to thank all of them, and of course, our collaborators who enable us to do all this research we love. Okay, thank you very much for your attention.
Okay, thank you, Miss Miroslava Harisova. Thank you. Okay, participant, before this simple seminar, I have a minimal knowledge about nanofiber technology, but before this seminar, I tried to learn little about this nanofiber technology, and I found many things about this beside the contradiction, but there's so many benefits from this nanofiber. And today, Miss Rafa Miroslav Farisova has explained us about silicon dioxide, cytosan, collagen, and silk fibroin. That I read that it's built from natural source, so it could sustainable source. And then that's why this is biocompatible and biodegradation is better than any synthetic material. It is, Miroslav Farisova?
Yes, that's true.
Okay. Okay, we have heard two amazing presentation from protein SARS-CoV-2 that so explained so detail with Prof. Jelly Nur Rahman and about the nanofiber, especially for medical approach, biomedical based nanofiber and their applicability in medicine. And then we'll continue with question and answer. Maybe before we continue, permit me, Prof, maybe even you have answer in the Q&A, maybe for other who don't read that, maybe you could try to explain more what the answer of any question. Permit, Prof, I will read the question and maybe you could help us to answer with with your speak, Prof. Permit us to open video, Prof. Could you open your video? Thank you, Prof. Sorry, Prof. Because it will be more spirit when we saw your face and you explain with your big spirit, Prof. It will communicable spirit. Okay, Prof. First of all, according to your explanation, there are so many phenotypes of COVID virus. What is the target of COVID vaccines in the virus so it can affect effectively? As you explained that virus contain from few non-structural protein and structural protein like envelope, membrane, nucleate, and what else? Envelope, spike. Maybe you could explain with attendance, Prof. Please, Prof.
Okay. Okay. Thank you. Okay.
This is from Dr. Dewi Nurfiana, specialist KFR.
Yeah.
Okay, Prof.
Okay. basically its protein of SARS-CoV-2 have a function in a virus. So the lack of activity of one protein may affect on the virus itself. So we can choose anything of each protein as a target of a COVID 19 vaccine. So this idea. Also, we can also design drug compound based on the certain protein structure. Okay. The question is which one of protein SARS-CoV-2 that more effective as a target? So the answer is a clinical test trial. So if we have target one protein and then we go to clinical trial, we can see the effective. So based on that. Actually we can use any protein SARS-CoV-2 as a target. People researcher right now interest in spike protein because this is the first protein that will interact with the receptor. So it more have spike protein more have attention than the other SARS-CoV-2 protein. This is understandable because when virus attack to the host cell,
Receptor.
it should be bind to receptor. So we have to inhibit the binding of spike protein to the receptor. This is a simple idea.
Simple
But
idea. Okay, bro.
but okay, but we can use any kind of protein because sometimes we need virus to infect ourselves, but with not pathogenic virus. So, but what we call virus yang ramah, because if we introduce that virus or something like that, we it can be
Harmful. You could be
improve
harmful from.
improve our immunity. That's my answer for
Okay,
Dr.
thank you,
Devi.
bro. Thank you, Prof. Dr. Devi has maybe Dr. Devi hope could and could understand with could clear with your explanation that actually spike protein is more interesting to be target of vaccine, ya, Prof.
Yes.
Because this is the site where COVID bind with the receptor of mucus. That's why maybe the other structure just like structure non-structural protein and structural protein others could be target of vaccine depending on the development of the SARS-CoV-2, ya, Prof. Okay, Prof. Thank you, Prof. And second, Prof, maybe before we go to next question for Prof. Jerry, maybe I could give the question for Mrs. Miss Lova. This is I want from anonymous, Mr. Lova, Miss Lova. I want to ask something, Miss. Why this new biosensing method using a nanofiber membrane is more favorable than conventional ELISA? And what kind of risk from this technology? Thank you.
Okay, thank you for the question. The application of the nanofibers in biosensing in comparison to nanoparticles can be based on their increased safety, because with nanoparticles, which can be also unstable in contact with proteins, the nanofibers can form clusters, can grow from nanoparticles into microparticles and change the sensitivity of the assay. This basically doesn't happen with the nanofibers because the structure is more stable, more rigid, and yet the surface available for the linking of the enzyme is very high. But usually the nanofibers, they don't cluster, they don't shrink, and don't change the surface which is available for the conjugation of the protein or any other biomolecule which is absorbed or conjugated into its surface. This is actually the reason why the nanofibrous technology is not used nowadays only in the biosensing and medicine, but also in water treatment. And it's the stability of nanofibers.
Yes, I see. Yeah, that's right. That's right. Yes. Nanofiber is more than only new biosensing method for nowadays, yeah, Miss Masifilova. So because it's more biocompatibility and biodegradation, I think, because this is from natural source, it's more and it's not make change in human body. That's why it could make in medical as medical treatment too. That's I saw from your first presentation. It has used for anti-infection, antibacterial agent, just like for Staphylococcus aureus and Pseudomonas aeruginosa. Even you use it for the other infection. Thank you. Maybe for the anonymous, this is enough. We go to next question. Prof. Jelly again. This is from anonymous attended, but maybe I will combine with from Dr. Agustias Ciptaningrum. For the first question, how amino acidic chain stability analysis suggests both mutation of the protein structure, and I have read that SARS-CoV-2 have been mutated. There are several forms of mutation. So how do the drug would be developed for this? Maybe call it how do vaccine could be developed if the SARS-CoV-2 is so easier for mutated. And you have said that there is a activity site from the SARS-CoV-2 protein. Maybe you could explain us from the many kind of protein that building the SARS-CoV-2, where's or which protein who is high risk for get activity site? Thank you, Prof. Please, Prof. About how amino acidic chain stability analysis suggests both mutation of the protein structure and that how SARS-CoV have been mutated many times. There are several form of mutation. So how do the drug would be developed? Even for my mind, there is not mutation, but there's many strain new of SARS-CoV-2. Okay, Prof. Please, Prof.
Okay, thank you. Okay, there is no rule of the specific amount of amino acid residue that may change stability of protein structure. So maybe only one mutation that can change the structure. But sometimes, like hemoglobin and like hemoglobin, this is both the similar protein that have similar function to transport oxygen. Hemoglobin have a function to transport oxygen in human being, but like hemoglobin have a function to transport oxygen in the root of tree. So the homology of that protein, the only 23%, but the activity and the folding is remain unchanged. So even the mutation is as much as like hemoglobin, but the folding and activity still exists, this is no problem. Another extreme example is sickle cell anemia. There is the only one mutation, glutamate residue in the surface of hemoglobin mutated to valine. Glutamate has hydrophilic 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 properties, but uh, the valine have a hydrophobic properties. So the mutations makes the single molecule of hemoglobin is unstable.
That's okay. And how about the drug would be developed, Prof? Have you an idea more for this?
Okay, if your target is active site of the protein, if the mutation not change the active site, so the drug can be active in the mutated protein. But there's problem if you develop vaccine, because the antibody recognized on the surface. So if you have mutation on the surface of protein, and the antibody may cannot recognize anymore. So, but if you develop uh, antiviral compound based on the active site activity, as long as the active site is not changed, okay, the compound can still inhibit even the protein, the protein already mutated. Thank you.
Okay. So if the is as long as the mutation is not occurred at activity site of the SARS-CoV protein, the drug is still could use for this SARS-CoV-2, Prof. Maybe
Okay,
I
exactly.
have yes, I have read many journal article that declare that SARS-CoV-2 is easier mutated viral. That's why. Maybe in the next in the future, Prof. Jelly and Mrs. Mislova could building a kind of nanofiber agent who could bind with the receptor of the viral SARS-CoV-2, and then SARS-CoV-2 couldn't bind to anything in our body, so it will be not only vaccine but drug, and okay, it will be the protective agent for the next COVID and any viral, because the viral always building a mutation in the body and usually it continue to be a cancer. Okay, next question for Mrs. Mislova. You have answered this, but maybe you better if you explain with your beauty explanation, Miss. Okay, from Bayu Irawan, Bayu Aryawan, Ahmad Bayu Aryawan, Faculty of Animal Husbandry, Halu Olea, University of Kendari, Mom Miroslava, silica was using in product for minimize degradation, which is being drying to storage the product. Will it inject many microbes if takes in open place a few minutes and continue to fresh or refrigerator secures? Because I have read from your explanation, Miss, there's different duration between silicon dioxide 1 hour, cytosan until 72 hours, collagen and silk fibroid. Maybe you could explain by the agent. Okay, based on the agent, Miss.
Yes. Uh, the thing is that the degradation we tested was tested in simulated body fluid. So it's basically degradation after it's put into the contact with your body, into the wound, or it's implanted for tissue engineering. All of the nanofibers I presented today are stable in the dry state. So basically, what we do before it goes into the in vitro tests or in vivo tests, the nanofibers are electrospun, then they are sterilized, closed into the sterilization package, and stored in, as mentioned, freezer or refrigerator in the sterile state and they don't degrade, basically. The degradation starts when it comes into the contact with the moisture, water, or the body fluids. Of course, the degradation in the in vivo state is accelerated because your body fluids and tissue contain different proteolytic enzymes which are able to decompose basically any scaffold you implant quite faster than when it's degraded only in the simulated conditions in vitro. And with the silica, I just want to mention that also the degradation kinetics in vitro or in vivo can be just like in the case of silk fibroin. They can be designed according to your need through the thermal stabilization of the nanofibers, basically. So there are some types of silica nanoparticles in general which are completely non-degradable due to high temperature of the stabilization. We are not trying to achieve this. So that's the reason why our silica is so quickly degraded, because we don't use the high temperature stabilization. We want them to degrade and release the salicylic acid. So I hope this response is enough.
Thank you, Ms. Miroslava, because we have limited time, but maybe this is the last question for Professor Jelly and last question for Mrs. Miroslava. This is for the Prof. Jelly first. Prof, this is, I think this last two questions have similar explanation, because I'll try to read from Hari Nugroho Eko Surniyantoro and Julham Yamamoto, that they ask about how relevation binding between SARS-CoV-2 with protein receptor. If the protein receptor get mutation, or how to make the protein receptor mutated so the SARS-CoV-2 protein, especially spike, couldn't binding to the receptor protein. Maybe just like this, maybe I read the question, Prof. Professor Nurrahman, very nice presentation. I would like to ask you, are there any possibilities if someone get the genetic polymorphism in their S2 gene? You explained that in SARS-CoV-2, the receptor is S2 gene. They'd be more resistant to the COVID infection. Thank you, Prof. From Hari from Badan Tenaga Nuklir Nasional. I think it's similar with from Julham Yamamoto. I'm Julham from University Sumatera Utara. I would like to ask a question to Prof. Jelly in order to avoid SARS-CoV-2 binding to its receptor. Is there a way to reduce the expression of S2 or to modify the S2 protein translationally? Because you are a protein expert, Prof. Maybe you could explain this. I'm sure you could explain this. Thank you, Prof. Time's yours.
Yeah. Okay, thank you. Thank you. I will first I will answer the simple question, but I cannot answer because for a question from Dr. Hari Nugroho Eko Surniyantoro from Badan Tenaga Nuklir Nasional. Actually, I do not have experience with working with polymorphism. So actually, I cannot answer even this simple question. But actually, honestly, I don't have experiences, so I cannot answer this question. But this from the first question from Zulham Yamamoto. The question is, in order to avoid SARS-CoV-2 binding to its receptor, it is in there a way to reduce the expression of S2 or to modify S2 post translational. Okay, I would like to answer why we have to reduce the expression, because S2 we can find on in all of our our cell receptor, not only in respiratory tract, also in other cells. So so uh, so if you want to avoid SARS-CoV-2 binding to its receptor, just put a molecule that can inhibit the binding of SARS-CoV-2 to receptor, uh, rather than you reduce expression of ACE2. This is my
Just
simple
like
answer.
kayu putih oil, Prof. Kayu putih oil or
Yeah,
essential other
yeah,
oil. And next maybe
yeah,
Miss Roslava will building nanofiber oil. So could inhibit binding of SARS-CoV-2 to the its S2 receptor. I think so. And I think she will build this after this presentation. Okay, Prof. Thank you, Prof. I understand. So if someone asks me about what is benefit of essential oil, just like kayu putih oil, I will explain just like what you explained me today, Prof, in protein knowledge aspect. Okay, for last question
yeah.
for Miss Miroslava from Czechia Republic. Is far from here. Thank you to get here, Miss. Okay, for the last question for Mr. Miss Lova, it's possible. Oh, yeah. Is it possible to develop nanofiber technology into drug slow release delivery system in order to ensure the bioavailability of the drug due to its stability? And maybe we combine with the last question. Silica is the product which was using as a gel sachet like silica gel for minimalized degradation, which is being drying to storage the product. For example, in shoes, food, or other. Will it inject many microbes if take in open place a few minutes or take to freezer or refrigerator for secure it? I'm sorry if you don't understand. And maybe he declared for the last question. Maybe it's not need to answer, but maybe you could answer from Muhammad Iqbal about this question. Please, your Mrs. Miroslava, for last question. Before we closing statement, prepare closing statement from Professor Julie and from Miroslava after this. Please, Mr. Miss.
Okay, thank you for the question. Yes, of course, it is possible to develop nanofiber technology into sustained or slow drug release. Actually, there are several technologies which are applied nowadays, uh, and you can choose the way of incorporation of the drug to the nanofibrosis system according of the kinetics or quickness of the release you are willing to obtain. If you conjugate or not conjugate, but absorb the drug on the surface of nanofibers, you obtain very prompt release and immediate action of the drug. This can be beneficial with some antiseptics when you want very quick interaction with the bacterial, or and it can be combined with a sustained release, so the action is very quick at the beginning and takes longer for several hours, several days. If you want postponed release and prolonged release for longer time, there are two main ways you can obtain this. And both of them were extensively studied up to the date. You can incorporate the drug into the nanofibers, into the matrix of nanofibers. Then it's released through the degradation of nanofibers or through diffusion from the nanofibers. But the possibility of incorporation is very limited by the solvent system you use for preparation of your nanofibers, because it doesn't necessarily be in compliance with need of the drug you want to incorporate. And one other method to obtain sustained release of the drug is using so-called core shell electrospinning or cold shell nanofibers. When you have a core of the nanofibers, which is usually filled with polymer, which is water-soluble, polyvinyl alcohol, polyethylene oxide, and there is the pure drug or mixture of water soluble drugs in the core, and the shell is formed of a more stable material like chitosan, for example, silk fibroin or polycaprolactone, and through this combination, the release of the drug is slowed down, so it can be sustained up to days or even weeks, depending on the solubility of the drug in the body fluids and the place of implantation of the way of introduction.
Okay. Thank you so much for Professor Jelly and for Miss Miroslava Ristova. Maybe we will meet in next meeting, maybe in another discussion. Maybe I'm as a physiologist from University of Lampung will be always prepared if you want to building a joint research or joint project. Maybe it would be interesting project for us, and it will bring many benefit for our institution. Okay, before we closing this session, may I hope you both first, maybe Professor Jelly could give us closing statement, please, Prof.
Yes. Okay. Uh, yeah. In my concluding remark said that to obtain SARS-CoV-2 drug, many people just thinking that uh, on the protein and genome. But if we go back to the pandemic in 1918, this occurred in Spain for avian flu, and the drug that effective for uh, uh, for uh, for uh, for treating avian flu is being based on glycan molecules. At the moment, nobody, not many researchers interest working with glycan uh, to glycan-based molecule to develop a drug. For example, zanamivir is a drug for avian flu that and the structure is based on salicylic acid. Salicylic acid is found in colostrum in the middle. So sometimes we can open a new research by searching of glycan molecule that can effectively inhibit SARS-CoV-2. Since many surface molecule of SARS-CoV-2 is proteoglycan. So this is open research that you can find glycan molecule as potential SARS-CoV-2 drugs in the future. Thank you.
Okay. Okay, proteoglycan is the point. I saw that from your explanation, protease and proteoglycan is a kind of protein that many in SARS-CoV-2 and could build many approach for this treatment, Prof. Thank you, Prof. Okay, for the last closing statement from Miss Miroslava Risova. Miroslava, sorry. Your name is so unfamiliar for Indonesian people, Miss. I hope you understand.
Yeah,
Okay.
I understand.
You have to change your name to other I'm familiar.
Okay,
Okay, Miss
yes.
Miroslava
Yeah, no.
Risova. Okay. Before you closing statement, I hope both of you do not close will not close the Zoom because the screen because our again we have Mr. Kholis Abdurrahman would like to give some few speak. Okay, please, Mr. Miroslava Risova for close statement.
I would just once again like to thank you for inviting me to this occasion. Uh, accord, as you can see in my lecture, nanofibrous structures have some potential in the medical application, in drug delivery and tissue engineering. But of course, just like every other material or nanomaterial, it has some risks which needs to be exploited, and we shouldn't rush to the conclusions too quickly. It's always good reason to test the materials properly, and that's basically why we are scientists, because we want not only to create but to explore and extend the knowledge, not only which is ours but of our students and colleagues also. So thank you very much.
Thank you so much. I couldn't wait to next project from Prof. Jelly and from Miss Miroslava with me. Thank you so much. And before that, I want to ask Miss Miroslava, what's time in your place, in your location? Could you give us the time?
It's 5:43 in the morning.
Oh my God.
Oh.
It's early morning. It's early morning. Very early morning. Thank you for your time. Okay, Miss Holis Abdurrahman, time is yours to give last last notification for this last statement for this our guest, invited speaker. Please, Prof.
Thank you very much for Dr. Nisa for the time. So I just want to briefly thanks especially to Prof. Zeli and also Dr. Mirka. I call her uh, Mirka. So thank you. Really, I really appreciate your time. I know this is very early in the morning in the Czech Republic, but you made it
Sacrifice, Prof.
perfectly. So really appreciate that, and we hope that, as Dr. Lisa mentioned, we can establish our future collaboration with UL Liberec. I spoke to Dr. Alena. Unfortunately, she couldn't make it today, but actually, I really want you both give a talk today. But yes, you really nailed it today, Dr. Mirka. So I really appreciate that, and hopefully, this can give us pave the road for our future collaboration, both with SGU, ITB, and also, of course, with the Unila in Ampung. So
Thank you.
we look forward for our future collaboration. Thank you,
Thank
Dr.
you
Reza.
for you
Thank
too. Thank you for you too, Prof. Hollis, before because you have many contribution for this seminar. This is our first international conference of agromedicine and medical science 2020 seminar, and it's so grateful for us too. Thank you so much for the Professor Holis Muhammad, uh, who Abdurrahman, who are bring many contribution to make this happen for our institution. And thank you so much for our invited speaker, especially
you. Yes, sure.
Mr. Mirka, Miss Miss Dr. Mirka and Prof. Jelly. Sorry, I call you Mirka. It's more easier for me.
Okay.
Okay. Thank you for your participation and contribution to give us many information and knowledge about this. Uh, as a moderator, thank you so much. I apologize if I make a few, maybe many mistake. Sorry, and with to Allah, I have apologies too. Thank you for your attention. It's finished for today. Maybe I hope we will get next collaboration, next project, and anything we could do together. Thank you so much. Thank you. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh. Good day all. Tabik
Thank you
pun,
again.
Mr.
Thank you so
Mirko,
much. Yeah.
you have say tabik
So
pun
you
iya
now
pun.
can have a rest, Mirka.
Yes.
Okay.
Take a
What,
rest.
I'm
what
sure
should
you're
I
awake
say?
the whole night.
Sorry, Mr.
What should
Mirka.
I say? It was tabikur,
Tabik
Pun.
pun, tabik pun.
tabikun.
It is Lampung. You Lampung regarding salam. Labu.
Oh,
Yes. Just if I say
Greeting.
tabik pun, Prof. Jelly, Prof. Holis, you have to answer iya pun.
iya
Iya
pun.
pun.
Yeah, pun.
Ya
So,
Yeah.
Mr.
Iya
Mirka,
pun.
Yeah.
you have to answer iya pun. Okay, I
Don't
repeated
worry,
my
Mirka. Even
verb.
myself don't understand that because that's a
Okay.
local language.
Yes. Okay. Okay. I repeat my salam. Tabik pun.
Yeah, pun.
Ya pun.
Thank you for cooperation.
Thank you
You
for everything.
next
Yeah.
next
So
video.
Okay.
Thank you so
actually,
much.
if you have
Thank
Bye-bye.
time,
you.
you can
I'll
stay
back
until
to
the closing
moderator.
ceremony.
See ya.
Thank you. Thank you.
Okay.
Bye
Thank
bye.
you.
Bye
Bye.
bye.
Thank you so much for our incredible invited speaker as well as our moderator. We already heard the conclusion of this first lecture from Dr. Khairuni Sabrawi, Asket MKes IFO. Next, we will have a closing speech from the Academic Dean of Faculty of Medicine, Lampung University. Please welcome Professor Dr. Muhartono, Asket MKes SPPA.
Assalamualaikum warahmatullahi wabarakatuh. Alhamdulillah, pujian bagi Allah. Kita memujinya, kita memohon pertolongannya, dan kita memohon ampunannya. Alhamdulillah, kegiatan ICAM ini telah berjalan dengan lancar. Seminar internasional ini diikuti oleh 1.100 peserta dari seluruh negara ya, kurang lebih ada 10 negara dan 90 presentasi oral. Saya ucapkan terima kasih kepada Bapak Rektor, Ibu Dekan, dan Ketua MABI, Ketua PBB MI, seluruh panitia ICAM 2020, panitia Dies FK Unila, dan Goodwood Publishing, dan seluruh peserta ICAM 2020. Semoga kegiatan ini dapat bermanfaat bagi dunia akademik. Dengan mengucapkan Alhamdulillah, kita tutup the first ICAM 2020. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Professor Dr. Muhartono Asket MKes SPPA for the speech. Next, we will move on to the photo session with all of the participants. To the committee, please take the picture.
Payah banget kemarin itu.
We have reached the end of our event today. Thank you, Dr. Asep Sukohar, Edkes MKes, and Dr. Miroslava Rishova as our invited speaker.
And we would like to thank you, Dr. Khairuni Sabrawi Asket MKes IFO, and Ms. Selfie Mawati SSE MSJ as our moderator, and also thank you for all of the participants.
See you at our next event. Thank you.
Tabik pun.
Ya pun, thank you.
Siap, Miss Mirka. Thank you, Prof. Jelly. Waiting for your project.
Have a good rest now, Mirka.
Have a nice rest, Miss Mirka. Have a nice dream.
I was supposed to visit Liberec this month, but due to COVID, I couldn't make it. So hopefully next time. Yeah. So Alena invited me to come there, but it's just nearly impossible to go there. Yeah. So.
But you could still make connection with Zoom or webinar or any kind mode information